Good morning, everyone, and a very warm welcome to Melbourne. I'm very glad that our speakers are all here, despite volcanic dust. And I'd like to welcome you all to this opening panel for this marvellous Festival of Ideas, for which we thank the University of Melbourne and Patrick McCackey. It's going to be a very exciting week, launched with great distinction last night. We were privileged to hear from Matt Ridley. I'm glad to see you here this morning, bright and early, Matt, as well. It was a really fabulous lecture. Thank you very much. So we're, of course, this morning going to be discussing the impact of the genetic revolution on human identity and health. I'm Suzanne Corey. I'm currently the President of the Australian Academy of Science and privileged to be a Vice-Chancellor's Fellow at this university. And I'll be chairing the session, and we're going to be hearing from three very dynamic speakers. Doug Hilton will be giving us an overview of the development of the genetic revolution, all the way from prehistoric agriculture to Mendel and his peas, from Watson and Crick to the Human Genome Project. And he's going to tell us how humans have actually used genetics over millennia to improve their lives. Vanessa Hayes, who's come all the way from the US to speak to us this morning, will describe how genomics is illuminating human evolution, our origins, diversity and migration out of Africa. And Brandon Wainwright will give us a glimpse of how genomics is transforming medicine and its prospects for enabling humans to live longer, healthier lives in the future. But before we hear from our speakers, let me just go back to basics and set the scene for you, because I know that many of you in the audience are not geneticists. So please excuse me, those of you who are very familiar with this stuff, but this is just to get everyone on the same page. So as I think everybody in our society is now aware, the stuff of life, at least for most organisms, is DNA. And in whoops, that was not the right slide thing, there it is. And in 1953, Francis Crick on the right and Jim Watson on the left discovered the structure of DNA, and in doing so revealed the fundamental secret of life. Here you can see them looking at that molecular model of DNA that they just completed. Two polymers wound around each other, rather like a twisted ladder, to form the elegant double helix, in a more elegant form on the left, that's rightly been called the Mona Lisa of modern science. Your DNA, quite literally, is your instruction manual. We call it your genome. Every cell in your body contains a complete copy of your genome, packaged into 46 chromosomes. Here visualized in a scanning electron microscope, these chromosomes. The DNA in every chromosome is wound up very tightly in coils. Indeed, if we took out the 46 DNA molecules from a single cell, unraveled them and placed them end to end, they would stretch for 1.5 meters. A staggering fact when you realize that a cell is so small that you have to look at it under the microscope. Each DNA strand is built from just 4 simple chemical entities that we call nucleotides, A, G, C, and T for short. These are the genetic letters, and it's the order or sequence of those letters along each strand that encrypts the genetic message. Critically, the two DNA strands are complementary. Each rung of the helix consists of interacting nucleotide pairs. A always pairs with T, and G always pairs with C. And this creates a molecular Velcro. So here you can see the molecular Velcro in action. The A pairing with the T's and the G's pairing with the C's make sure that these two DNA strands zip up together. As Watson, no, the important point about this, as Watson and Crick so laconically, if you like, pointed out in their very famous paper, is the complementarity of the strands that immediately explained one of the deepest mysteries of biology, how genes can duplicate themselves and be handed on from one cell to two daughter cells. Because as the DNA strand replicates, the new strands are built up with A's and T's, and so they're faithfully replicated. And so we end up with two identical DNA helices in each of the daughter cells. This process is incredibly accurate, but not quite perfect. And every now and then, mistakes creep in, and that creates the mutations that evolution thrives upon. So let me conclude this introduction by just showing you the amazing molecular machine that accomplishes this critical task. This has been created by Drew Berry from the Walter and Liza Hall Institute in as accurate molecular detail as we know at this point in time. It's really quite remarkable. So this is how DNA replication occurs, how the two strands are copied into identical helices and passed on to the two daughter cells. Here we go.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So I don't know about you, but whenever I see this animation, I simply marvel at the miracle of evolution and stand in awe of the molecular machines that sustain my life. So let's move on, and let me introduce you to our first speaker, Professor Doug Hilton. Maybe I have to move this. Yes. Professor Doug Hilton is Director of Melbourne's Walter and Eliza Hall Institute. He works on the genetics of blood cell formation and has made major contributions to understanding cell signalling. Today, as I said before, he's going to give us a sweeping view of the history of genetics, and the title of his talk is The Great Genome Journey, an odyssey of underestimated difficulty, overestimated tragedy, and remarkable benefits. Doug, welcome.
Sure. Well, thank you very much. It's actually a really great pleasure to be able to give this talk. It's not a talk I've given before, so if I stumble in parts, then you can forgive me. I'd also like just at the outset to thank Vanessa Solomon, who's worked with me to put together this talk. I think that's an important acknowledgement. Often, I think we associate the Genome Project with a single landmark, and that was a very unusual press conference that took place almost 11 years ago to the day. And it took place in the White House, and it involved people you might not normally think of as giving press conferences about scientific breakthroughs. It involved Bill Clinton and via satellite Tony Blair. And Bill was flanked by two scientists, Francis Collins from the National Institutes of Health and Craig Venter, who was running a biotechnology company called Celera. And both of those groups were partly racing, partly cooperating to put together the human genome. And I'll come back to what that actually meant. What they announced was that they had created the first draft sequence of all of those letters that Suzanne introduced, all of the letters of DNA in a human cell, all of those letters being the human genome. They created this draft sequence and announced it to the world. And what I thought was really illuminating were some of the quotes that came out of that conference. It was obviously a time of great hope and in many ways great hype. This is a problem. There it is, 3 clicks. The first quote, and I could have chosen any two of dozens of quotes from the leading protagonists here, as well as others commenting on the field. The decoding of the genome will change the self-understanding of humanity and will be one of the strongest driving forces of the world economy. That was actually midway through the Genome Project, a couple of years before the press conference. I also think Francis Collins' quote was interesting. This is taking multiple clicks, isn't it? Sorry. I think it's interesting because he even puts a date on when the breakthroughs are going to happen. By 2010, the field of pharmacogenomics will have blossomed, and a prophylactic drug regimen based on the knowledge of a patient's personal genetic data will be precisely described. So this is really a vision where you or your Auntie Maud or one of your children will have their genome sequenced. They'll go into their general practitioners with a DVD or some sort of disk drive that has the sequence on it. The GP will look at the sequence and will tell you not only the things that you can do to prevent the illnesses that you're predisposed to, but also will be tailoring specific therapies to help cure your personal type of diabetes or cancer. And as I'm sure Brandon will talk about at the end, and I'm going to mention at the end, cancer is probably where we're coming closest to living this vision at the moment. What I'd like to do is to really convince you that far from being a single breakthrough event, our understanding of how our genes affect our characteristics and that's really what the Genome Project is about. How do our genes that we pass on to children that are inherited from our parents affect our traits and characters? That our understanding of that has taken a very long time. And in fact, the reality of the journey is more exciting than the hype that surrounds a particular event. So if you strip away the hype, and if you also strip away the doom that we've seen from a number of commentators when they make public pronouncements or in a number of different dystopian novels, if you strip away the hype and the doom, the journey is actually a lot more interesting. It's a journey that's taken millennium. It includes some of the most exciting scientific discoveries that humanity's made. It's involved a whole host of players. It's occurred small step by small step, and it's inexorably delivered a lot of benefits that we now take for granted for health, for agriculture, for a range of things. So what I'd like to do is to outline what I think are the 5 key landmarks of that journey. I don't want to be dogmatic about them. I want us to think about where that journey began, what were the key milestones, and then perhaps at the end reflect on where we may be heading in terms of that journey. Maybe the first, and to think of that first landmark, we kind of have to put ourselves in almost a prehistoric mindset. Probably, you know, my thoughts around what the first landmark would have been would be the realisation that characteristics are inherited. That is, that offspring resemble their parents more than they do the general population of that species. To me, that would be the first conception of things being heritable. I guess I have a little bit of an issue with separating the first and the second landmarks in the story, and that's all I'm going to say about the first landmark. I wonder actually whether there was a time where we realised characteristics were inherited, but we actually never used the information. Was there a point at which it was simply theory or observation, and that observation wasn't put into practice by the people that had that realisation? So the second step, I think, is that those characteristics that are inheritable are selectable by man for benefits. And then obviously, the second step was the realisation that those characteristics were also selectable by nature. Perhaps the next step in this long journey was the realisation or understanding of the rules of heredity. And that's a process that we're still in the midst of. It may seem like everything was worked out with Mendelian genetics, as I'll mention through the talk, but I'd like to also point out that we're far from understanding the rules that lead to inheritance of complex diseases that are a mixture of a lot of different genes and the environment. Obviously, the 20th century has been a century where we've examined the physical basis of heredity, and Suzanne has talked about that. And then I think the final step is really how we begin to overcome our heredity. So let me go back. And I think there's ample evidence from the archaeological record and from molecular biology looking at different breeds that have arisen and been selected for in different parts of the world, that really hand in hand with domestication and agriculture came the ability of farmers to select traits of animals and plants that suited their purpose. And if we look at this image from 2000 years BC, we can see at least 3 different recognisable breeds of dogs that had been selected by Egyptians even nearly 4,000 years ago. So I think there's ample evidence that throughout the last 5,000 to 10,000 years, farmers and other people have been selecting for traits that they were most interested in. Really, there was then a 3,500-year gap in a sense, and that was until Darwin realised that traits could also be naturally selected. And there was varieties of that, earlier varieties of that sort of thinking through Lamarck and Darwin's grandfather. But Darwin, interestingly, spent the first 2 chapters of his book, 59 pages, outlining the evidence for artificial selection, that is, the selection of different breeds of animals, because the weight of evidence for that was so overwhelming, he thought it best to start with something that was uncontroversial and really obvious to people in Victorian England. And he then went on to describe the idea that differences in individuals within a species were selected by nature to lead to the much more tumultuous conclusion that species arose by natural selection. Again, there was a little gap, and while the farmers were really the first applied geneticists, Mendel was the first scientist to be able to begin to understand the rules of heredity. And I think Mendel's genius came probably in many ways from the same sort of characteristic that is typical of many great scientists. He was able to reduce what seemed like an intractable, complex problem to a simple system. His genius lay in the use of a simple experimental organism, the pea, and the study of characters which, rather than varying continuously, were dichotomous, that is, green peas, yellow peas, wrinkled pods, smooth pods, tall plants, small plants. And by reducing the problem of genetics down to those simple traits, he was able to work out that those characters were heritable predictably and was able to work out the mathematics behind it. Mendel's work was rediscovered about 35 years after he wrote it by 3 people, De Vries, Correns and Bateson. And I want to focus on Bateson because I was really struck by some of his statements. He was really an evangelical character. He was absolutely full of hope for the promise of genetics. And I was struck that a couple of quotes that are in his book of 1900 seemed very similar to the sorts of comments that were being made 100 or so years later by Venter and Collins. And again, I'd like to read these. An exact determination of the laws of heredity will probably work more change in man's outlook of the world and his power over nature than any other advance in natural knowledge that can be foreseen. I could actually imagine him standing next to Bill Clinton and saying that. And I think even better was the complete misjudgment of how long it would take. There is no doubt whatever that these laws can be determined in comparison with the labour that has been needed for other great discoveries. We may even expect that the necessary effort will be small. 100 years later, we're in the midst of a series of experiments called genome-wide association experiments, which aim to try and understand which of the many millions of differences in my DNA from your DNA contribute to the onset of common diseases like diabetes or obesity or any number of complex traits. I think that one of the interesting things is we look at these studies now, and one of the puzzles we have is a problem called missing heredity. We can only account for a fraction of the genetics that we know contributes to those diseases using these massive studies. So billions of dollars and 100 years later, I think we still have a long way to go to be able to work out the precise rules of heredity. So again, I think somebody with unbridled enthusiasm, but maybe just a tad too optimistic to get the reality right. Suzanne has talked a little bit about some of the steps that have led to our understanding of the physical basis of heredity. It was only a few years after Mendel's work was rediscovered that one of my favourite scientists, Thomas Hunt Morgan, found out that genes are actually found on chromosomes. That is, chromosomes contain the elements of heredity. The interesting thing about chromosomes is they contain two types of molecules. The DNA, which Suzanne talked about, but a big layer of proteins that cover the chromosomes, cover the DNA and wind them up tight. And there was a lot of debate about whether the DNA or the proteins were actually the substance of heredity, the material that passed on the traits from parent to offspring. And it took nearly 30 years, more than 30 years, before DNA was shown to most scientists' satisfaction to be the heritable material. The idea that genes encode proteins, and that's the mechanism by which characteristics manifest, was first proposed in 1902 by Garrod. And he was actually studying the human as an experimental system and was the person that understood that inborn errors of metabolism arose from mutations in genes that encoded single proteins. But again, despite that evidence, it took another nearly 40 years for that to become widely accepted and for experiments to validate that hypothesis. Suzanne's already talked about the landmark of understanding the structure of DNA, the central dogma that DNA encodes protein via an intermediate RNA molecule was postulated by Crick, and then within 8 years of solving the structure of DNA, the genetic code was determined. That is, we worked out what sequences of DNA encoded which amino acids present in protein. Really, the dawn of the Genome Project in many ways came with the sequencing of the first genes. So the genetic code was actually worked out by synthesising artificial pieces of DNA and working out what type of amino acids were encoded. The first naturally occurring genes were sequenced in a convenient experimental organism called a bacteriophage, and that's a virus that infects a bacteria. And that really wasn't anything to do with trying to gain biological insight. It was just the most convenient tool to produce DNA that allowed it to be sequenced. And likewise, the first human gene that was sequenced was placental lactogen, again, a protein that we're not particularly interested in today. So this was the first faltering steps of genome projects. The first entire genome to be sequenced, and you may be surprised to know that it happened nearly 40 years ago. And again, it was a bacteriophage, a little virus that doesn't have too many letters and was a simple starting point. It was then 20 years before, using the technology that was developed and automated as part of the Human Genome Project that we were able to then sequence the genome of a bacteria. The next cabs off the rank were all organisms that scientists were very excited about because they were genetically tractable organisms that served as a model for a lot of the processes that go on in the human being. And that is the yeast, Saccharomyces cerevisiae, a little nematode worm, and a scraggly little plant called Arabidopsis. And then at about the same time of that press conference, more medically interesting genomes were isolated. The mouse genome, which serves as the best model for human biology and disease, the parasite Plasmodium falciparum, and in 2003, a couple of years after the press conference, the human genome was completed, which meant they'd filled in most of the gaps and eliminated most of the errors and were reasonably comfortable that they had a good record. I like the fact that they had the press conference 2 years before they actually got to the completion. So where are we heading? It's been really staggering that in the 8 or 9 years since the completion of the human genome, in fact, we've probably made the greatest strides forward with technology. The technology that was used to sequence the human genome between 1990 and 2003 was exactly the same type of technology that Sanger used to sequence the first genome of that bacteriophage. It's called the Sanger method or the dideoxy method. And in fact, a lot of the ability to sequence the human genome wasn't technological changes in that method per se, but automation and throwing a lot of money at the problem. In the last period, from 2007 onwards, what we found is that there have been a number of revolutionary changes in the way DNA is sequenced that have driven the cost of DNA sequencing down inexorably, it feels. So it took nearly $3 billion in 13 years to sequence the first human genome. At the start of that journey, it cost about $10 per letter to get the sequence. By 2001, that cost had come down to about 10 cents per letter. And at the moment, the cost is that we would get 300,000 letters for 10 cents. So the cost of sequencing the genome has come down firstly as a consequence of wanting to do the Genome Project. So technology and solving the technological problems and automation really drove the cost down. But in the last 10 years, the cost has come down even further because of some really profound breakthroughs, to the point where we can now contemplate a $100 genome. So this is actually an ad from 23andMe, which is a US company that will sequence part of your DNA. And so I think this really does give us a glimpse of what the Emerald City might look like in a few years' time. I think it will be possible in a few years' time for most Australians to be able to afford to have their DNA sequenced. The question, and obviously the promise there is that we will be able to tailor medicines or preventions to your DNA. That is, we'll be able to treat different diseases as if they were individual diseases, not lumping everything in together. And we've grown a little bit used to that concept when we think about cancer. 40 or 50 years ago, cancer was viewed as a single disease. We've certainly got used to the idea of there being different types of cancer, depending on the tissue of origin, breast cancer, colon cancer. Over the last few years, we've become accustomed to the idea that not all breast cancer is the same. It's possible to stratify different types of breast cancer based on the cell within the breast that the cancer originated in, and also the molecular changes that led to the creation of the cancer. And it's certainly true now that by sequencing cancer genomes and understanding the precise changes that have gone on in individual cancers, that we can tailor therapies that tackle those individual problems rather than treating people generically with chemotherapy with all the side effects. So I think the challenge for us is now not a technological challenge, it's not a price barrier. I think what we have to do is to ensure that the general community understands what can be achieved with an individual's genome. But perhaps the even bigger challenge is to work with our clinical colleagues, work with the medical sector, to be able to train a generation of clinicians that are able to sit down with people, understand what their genome tells them, and to be able to tailor prevention strategies and therapies that suit the individual. So I think there is an Emerald City that is, I'm not going to make a judgment about when we will get to the Emerald City, but I think there is an Emerald City. I think there's an enormous amount of hope that's been derived from our experience working with cancers, and I'm sure that's going to be applied in the future to other diseases. So I think it's a fantastic time. It's really exciting to be a scientist. I think one of the great benefits of the Human Genome Project from a scientist viewpoint is we don't now have to spend months and years assembling the tools to do our experiments. Those tools have already been assembled for us. We can get on and do interesting experiments much more quickly than we could a decade ago, and I'm sure that's going to translate to benefits for the general community. Thank you, Suzanne. Thank you.
Thank you, Doug. Now, we're going to have lots of time for questions in the panel discussion following the lectures, so I'll ask you to store up all of those burning questions you had for Doug and launch them at him in our panel discussion. I also forgot to tell you, please turn off your mobile phones if you haven't already done so. So our next speaker is Vanessa Hayes, who's Professor of Genomic Medicine at the Craig Venter Institute in San Diego. Craig Venter was one of those illustrious geneticists you saw in Doug's first slide. As you'll see, Vanessa is a woman of incredible drive and energy, and her studies of African genetic populations in particular are totally transforming our understanding of human origins. The title of her talk, which I'm looking forward to very much, is Genomic Revolution, Human Evolution. What does it mean to be human? Vanessa, welcome to the platform. And to Melbourne.
Thank you. Leave it here. Thank you, Suzanne. And thank you to the organisers for inviting me to Melbourne and for making it so hard to get here. So I landed up in Sydney and had to drive. Anyway, I made it here. And thank you very much for having me here. And also, hello to everyone from Craig Venter. You've heard a bit about him this morning. Whoops. Okay, so you heard in the previous talk from Doug about the Human Genome Revolution. And in February this year in San Diego, we had a celebration of 10 years since the publications of the Human Genome. And the point of this celebration was really to look forward. What is in store for us for the next 10 years? And you heard that really it was the technological revolution that has made it possible for us to move from that $3 billion project. It took 13 years to complete. Just imagine, it was the same as putting man on moon, the equivalent of doing the first human genome sequence. So we're now at a stage where laboratories here in Australia, you'll hear from Brandon afterwards, can actually use these technologies. It wasn't possible in the past. There were only 4 centers in the world sequencing the human genome, and they had hundreds of machines. The Craig Venter Institute, for example, at that time, Celera, had 500 machines, 500 staff working on 100 machines. We are now down to 9 machines. So the technology is really driving the field, and the reason for that is we're generating terabytes of data. We can't deal with all the data. We need more brains and more people out there to actually deal with the data. And you'll see in the photograph again, Craig Venter and Francis Collins together with the head of the X Prize, with the goal to bring the price of sequencing even down, further down to at least $1,000 and even further, as you've heard from Doug, so that this technology can be available for everybody to revolutionize the field. But what I want to present today is how we can actually use this genomic technology beyond just looking at the human genome, but we can start looking back in time. We can start understanding who we are and what it is to be human. And of course, to understand that question is also to understand human disease. So this was the global map of human genomics in 2010. You'll also hear from Brandon how personalized genomics has come about. And what do we mean by personalized genomics? It means it's sequencing the whole DNA sequence, the genome, which is 3 billion of those building blocks, But of course, we inherit 3 billion from mom and 3 billion from dad, so actually we're 6 billion. And what the difference is, the Human Genome Project was not a single individual. The reason for that is we didn't quite know at that time how to deal with the ethics around having a single person sequence, putting the DNA sequence out there, and how that would impact that individual, knowing everything about them. I always say I have the best job in the world because I'm the only person who knows every single thing about my boss. And he loves it every time I sequence his genome. So it's really great. He can't hide a thing from me. But what it looked like in 2010 was basically a picture on the left. If you were grey-haired, balding, genomicist, then that was the human genome, the personal genome, because we first had Craig Venter in 2007, Jim Watson in 2008, George Church in 2010, and so on, so on. So if you were an American and a genomicist and European, then you put your genome out there. And they did this because of ethical reasons again. No one knew how to approach the ethical question. So if you're a genomicist and you put your own sequence out there, well, it's okay, no one can complain. So the world of genomics, with the 8 genomes that were out there, was mainly Europeans from America, and we had one African genome. And why is this important? This was an unnamed individual. It was a male from Yoruba, from Nigeria, a Yoruban male from Nigeria. And there was no face put to this genome because of ethical reasons, once again. But this was important because Africa is the birthplace of all of us. And there's not only 2,000 different languages within Africa, but it holds 1/3 of the world's diversity. So if we're really going to understand ourselves, we are more than just Europeans. And one could say the same for Australia. There are over 400 different Aboriginal language groups, yet there's not a single Aboriginal genome. So how can we think of ourselves as a world species if we continue down this route? So I'll just quickly touch on why Africa is important when it comes to genomics or understanding ourselves. It is the place we believe around 7 million years ago that we had a common ancestor with our chimp cousins. About 5.7 million years ago, the oldest hominid remains have been found within Africa. 2 million years ago, the genus Homo emerges also within Africa. About 500,000 years ago, the archaic homin species, the most common one we know about being Neanderthal, diverges within Africa. 200,000 years ago, modern humans, our common ancestor, diverged once again within Africa. We don't know where the spot of this divergence was. If you're an archaeologist, you're probably more inclined to think this was in East Africa. If you're a geneticist or recently linguist, you'll believe that this was in Southern African region. About 35,000 years ago, we know that our nearest cousins, the Neanderthal, went extinct. This, of course, means, because we migrated out of Africa around 50 to 70,000 years ago, that we must have been living side by side, these archaic species. And about 5,000 years ago, we had an important migration also within Africa. And migrations are important because they've driven genomic change within us. So this is the area of interest of my research team. And the reason for this region, it's called the Kalahari Basin or Kalahari Desert. It is home to who we believe is the oldest living descendants of modern man. And these are the Kusan people. The word Kusan, linguistically, means people who click. They click when they speak. If they do not have a click, it's equivalent to a consonant, then generally they are not a Kusan person. This is important because we believe that these people have been living in this region for at least 100,000 years. So they tell us about our history. So in 2010, we published the first not only indigenous genome sequence, but the first sequence for a Bantu individual. There are over 500 Bantu linguistic groups. Those are the non-click speaking people, as well as the click speaking people and in this case, Igubi. So the individual sequenced was Archbishop Desmond Tutu, as you hopefully all recognize, and this individual called Igubi, he's a click speaker from the Kalahari. And what did this tell us? Whoops. If we take Craig Venter's genome, out of those 3 billion building blocks, he has roughly 3 million differences, changes. If we take Archbishop Tutu, he has 3.6 million differences in his genome. If we take Igubi, he has over 4 million. So what does that say? It tells us that Igubi and his ancestors have been living on this earth much longer than Europeans and longer than the Archbishop's ancestors. So it tells us the amount of diversity that still exists in Africa. When we went further and compared 2 Ku speakers that live only 500 kilometers from each other today, both of them click, but they tell me we don't understand each other. We found more differences between those 2 click speakers who, to our eye, look identical, still hunt and gather. They're more different than I am from a Chinese person. You might as well call me Chinese. So that just shows you the amount of diversity we still had to capture within Africa. So in that project, we found 1.3 million variants that hadn't been found before. So we still needed to understand the complexity of our cells. We also found changes that were associated with 621 diseases. This is important. If we don't look at ourselves as a global species, we'll never understand what is truly disease-associated. Some examples, the Kusan speakers, for example, they're all lactose intolerant because we were all lactose intolerant. We were all hunter-gatherers. Around 10 to 12,000 years ago, we changed to agriculture. And then it became essential for us to become tolerant to drinking milk. But they are still all intolerant. They also don't have the resistance to malaria, for example. It's important for their survival because they're completely surrounded by malaria. Why aren't they? Because they don't have cattle, they don't store water. There's nowhere for the mosquitoes to breed. So these are important evolutionary steps we have had to adapt to survive. We are all survivors in this room. I just put this photograph up on the 3 genomes that we work with quite readily in our lab. We've got Craig Venter and the Archbishop Desmond Tutu. This was at our dinner at the beginning of this year to celebrate 10 years since the publication of the Human Genome. And how many differences do you think that Craig Venter and the Archbishop share? Well, they share 500,000 differences in their genome. But if we take the Archbishop and Igubi, who live very close to each other in Africa, how many differences do they share? They also share 500,000. And why is that important? Because when we are looking at designing pharmaceutical drugs that are going to be relevant for people, we need to understand what is actually going to be relevant for a specific population or a group. And we can't just assume that Africa is one place. So this is a global tree of diversity, if we take the entire globe. And you can see the out of Africa diversity and the within Africa diversity. And we haven't really captured the whole of Africa, and that's just the part of Africa that we have captured and we have got data on. And you see the amount of branching that occurs, and this very, very long branch that sticks out. That just shows the amount of diversity within the continent, and we still have to understand that. But of course, we also have to understand our migration out of Africa, because this was an essential event that has shaped the face of modern humans as at this time when we migrated out. And of course, Australia has an important role because it's believed that the Aboriginal Australians represent that first wave of migration around 60,000 years ago. And as I mentioned earlier, when modern man migrated out of Africa, they met up with their cousins, the Neanderthals. It was always believed until last year that no interbreeding occurred between modern humans and Neanderthals. As you know from the skeleton remains, Neanderthals are quite different. They were not cricketers, they could not bowl, Their arm got stuck here. It's got to do with their skeletal. We know a lot about the facial features, the high brow, the lack of chin. And we know that most Neanderthal remains have been found in Europe. So we know that we were living side by side them. Yet we assumed, and why did we assume there was no interbreeding? Because we were only looking at the mother's DNA. There's part of DNA that's only inherited by the mother, from the mother, and that's called the mitochondrial DNA. It gives 16,500 bases to their children. And the dad, I'm terribly sorry, you don't contribute anything to that, so you always give a little less. Mom always gives a bit more. And because we were restricting our analysis only to that maternal piece of DNA, because it's really hard to get DNA out of these bones, we were restricted in our analysis. So in 2010, and also, by the way, from this analysis, it was clear that I would be Neanderthal with my red hair. Neanderthals tended to be red-haired and very fair. So the end of last year, or June last year, Svante Pääbo from Max Planck in Germany, along with his colleagues, managed to sequence the Neanderthal genome. So that was not biased to mum anymore. It was what we inherited from both our parents. And what they found was that in all Europeans and Asians, we contain about 1% to 4% Neanderthal genome. So interbreeding did occur with our archaic cousins. It was through the male line. And this is important because remember, once again, that shapes who we are. So, of course, I went and looked at my boss. He's 3% Neanderthal. Looked at myself. He's 3%. I'm also 3% Neanderthal. I also looked at a very famous Australian. I didn't ask for permission to use his name today, but he's 6% Neanderthal. So I enjoyed telling him that the other day. But what was really interesting is this interbreeding occurred outside of Africa. So we did not find any Neanderthal sequence within Africa. But of course, what is really interesting now is the reason that we can actually do this research, the reason that we are able now to look back in time is really this technological advancement, that we are able to take DNA from the root of a tooth, or from a bone, or from a hair, even from nails, and actually extract the DNA and use and sequence that DNA. We couldn't do that using the old technology, but with the new technology, we are quite clever of actually doing that. And what happened also last year was the discovery of a new archaic species, the Denisovians. They lived, a finger, a little baby pinky finger and a tooth was found in a cave in the Siberia region, and it's believed that this species diverged from the Neanderthals about 1 million years ago. And what did they find by doing whole genome sequencing was that all Melanesians have 1% to 6% Denisovian DNA, but no one else in the world. So you see how we are also being shaped by looking at our cousins. So where are we? We can look back in time. We can look forward. We can now start understanding who we are. But we've spoken, both Doug and myself have spoken today about the human genome and about what we inherited. What I want to show from this slide and talk a little bit around it is that we are actually more than just what we inherit. So this further complicates the matter. So at the J. Craig Venter Institute, we are interested in using genomics as the tool to understand human life. You heard earlier that in 1995, the J. Craig Venter Institute sequenced the first free living organism, being the influenza, and in 2001 contributed to the publication of the first human genome. But as we said, that was only 3 billion bases of building block. So what we did in 2007 with Craig's genome was sequence what we call a diploid genome, that is the information from mom and dad taken into account, so 6 billion bases. In 2010, we sequenced the first African and indigenous genome. But what I really want to point out is this work in 2006 and this work in 2007. What this work illustrates is, as humans Sure, we inherit our DNA from our parents, but we have 10,000-fold more bugs living on and in us than we do our own DNA. And that's important. It defines who we are, it defines whether we get sick or we don't get sick, and how we survive on this planet. We know that due to the fact of sterilization, our flora inside of us is changing, and how we respond to diseases is changing. So in 2006 at the Venter Institute, we published the first gut, what we call microbiome. So Karen Nelson, our director, always jokes and says, while Craig was traveling around the planet on his, around the oceans on his boat, where he circumvented the globe for 2 years collecting sea samples, she was collecting feces samples. And what she did was then sequence from the feces of humans the microflora that lives within us. And it was astounding what we found and how many new species were actually identified. And this is really important to know what lives in and on us. But this was also important what Craig was doing. He wasn't just having fun, well, he probably was, but what he was doing was collecting seawater. And from those seawater, we couldn't, without the new tools of genomics, actually look at what's in our environment. Now we capture air, we capture sea, things that we cannot see with our naked eye. But using sequencing, we can identify what's in that, what we are breathing in, what we are living in. And that is further leading to our understanding of what it is to be human and how we survive with the challenges around us. Of course, we have to address a lot of ethical issues associated with the work that we do. And also in 2010, the Venter Institute was the first to generate a synthetic cell. And what does this mean? That means mom and dad was a computer, and from that, a genome was generated, 1 million bases, and it's surviving. It's still living in the lab today. So we are moving on in the field of genomics. What is the purpose of this last work? It took 15 years to complete. The purpose is to design vaccines, to be able to generate vaccines a lot faster in the lab than we currently can when something like the flu breaks out, and also to generate synthetic fuel because the planet is running out of fuel. So the tool of genomics is not only to understand who we are, but also to take us forward into the new world. Thank you very much.
Thank you very much, Vanessa. And now we move on to our final speaker before the panel discussion, who is Professor Brandon Wainwright. He's Director of the Institute of Molecular Biosciences at the University of Queensland. Brandon studies the genetics of complex inherited human diseases. In particular, he's famous for his work on cystic fibrosis and basal carcinoma of the skin. Brandon's going to take us into the future, when each of us will have our genome analysed at birth for $1,000, and he's going to tell us how that knowledge will help our parents and ourselves and have impact on our life's journey. Brandon.
Thanks, Suzanne, and thank you to the organisers for the invitation today. And Doug and Vanessa have given me an absolutely fantastic start to my talk. And actually, I noticed before in the run-through, I actually like the idea of being a writer and historian. I hope it pays well. That's the only thing. So what actually, both Doug and Vanessa referred to personalised genomics, and I'm actually not going to talk in one area of the application of genomics to populations, and that's in cancer genomics, for which Australia has a very large effort, and I'm happy to answer questions on it later, where we take cancer from individuals, determine their DNA sequence, try to work out what's going on, and then to tailor therapies. Rather than that, I'm going to take a slightly more controversial view, I think, and which will probably lead us into some ethical questions in addition to the ones raised by Doug and Vanessa. And that's to look at the application of genomics not to, say, specific disease states, but to a population and where that's going to take us. As both Doug and Vanessa have already told us, we as individuals are a combination of the genes we were dealt, the variations in our genome and our environment, and as we've just heard, the pathogens we may be carrying within us, and the way we interact with the genomes of organisms around us. We tend to think of disease, for example, and characteristics as we can go all the way in the spectrum from, say, a disease such as cystic fibrosis, where if you get the gene, you get the disease. There's a little variation around that. But basically, if you get the disease, you get the gene, all the way at the other end of things that we think of as perhaps environmental, but the twin geneticists tell us there's a tiny little bit of genetics in there, like voting habit, which has got a tiny little component of genetics, small, but most of it's environmental. And all of the things that we imagine that define us as individuals are somewhere in that spectrum, the balance between the variation in our DNA and our interaction with the world around us. So in order to illustrate this and where we may be heading, I've actually taken the easy route out today, and I'm just going to talk, it's light on here but dark on there. I'm actually going to talk about a movie called Gattaca, and you would know already that these are the letters of DNA sequence, G-A-T-A-C-A, and you can just make out there. So Gattaca it's actually an interesting movie because it was filmed before the human genome was sequenced. And so it's a predictive of a future Earth, where at birth, and here we have the hero of the story, at birth, there's the doctor about to stab the hero with a needle and pull out a drop of blood, and that goes straight into a machine. Click. And from that machine, the sequence is determined at birth. And the doctor, at this point in the movie, is reading out the DNA sequence within a couple of minutes, and the story goes something like 40% chance of neurological disorder, 60% chance of attention deficit disorder, 80% chance of manic depressive illness, 98% chance of heart failure, predicted lifespan 30.1 years. Now, this is funny, right? So, maybe not. So, the movie then goes on. So our hero is therefore consigned to some kind of position in life as a janitor, as we'll see. Now, his parents, on the basis of his poor genetics and poor expected contribution to society. Now, his parents then go for their next child, for a more conventional route, and that is to go through the in vitro fertilization route. And in this scenario, the IVF is done. A cell is taken from individual embryos. The DNA is sequenced. The geneticist here looks at the genome and sits down with the parents and says, well, you've selected a child with blue eyes, curly hair, IQ in the following range. We can do all that. And I've taken the liberty to correct a few genetic defects I saw in there. And so, you can have this one or this one, and this will be exactly what you want. And of course, the parents who had conceived our hero, John, earlier, as in natural circumstances, he therefore became known within the movie as an invalid. This person, this fetus who's about to become a person, is a valid because their genetic composition has been determined and selected. The parents wanted to mix it up a bit and said, well, can't you just leave this alone? We'd like to take our chances a bit. We've selected a boy and the blue eyes and things like that. But what about, let's just leave the rest. And I think there's a great quote from the movie here where the geneticist says, it's still you. It's just the best of you. So it's taken, the IQ is in the range between the two parents, the predicted IQ and, all these other things. It's still the parents, it's just the best of the parents. And I think that's an excellent quote. So, we go on. So here is our hero, John, and he's in his position, Ethan Hawke, for those of you who are looking for movie stars. And so he's in a rather lowly position within a space center, and he aspires to be an astronaut. So what he does is, the tale here is that he has bad genetics, but I'll give you the end of it now without giving away the twists in the plot. The whole point of this story is that he overcomes his bad genetics because nothing is absolutely determined at birth. There's always, in this case, a small environmental component. So even though society had determined him a particular way, he ultimately triumphs over his environment. So in order to achieve this and get into the space program, this is Jude Law. So Jude Law is a perfect specimen, but unfortunately, one of the highly selected individuals who sells his identity to John, our hero, here he is giving blood, because in this society, just to walk through, instead of swiping a card, put your finger on it, and the DNA is determined, and your identity pops up. So everywhere you go, you're defined by your genetics. But I guess the tale in here is that he's actually paralysed because going along with this high level of performance is a degree of risk-taking, and he took one risk too many and fell off a cliff and became paralysed, and then decided to sell his identity to our hero John. And so using blood, skin scrapings, hair, urine samples, and the provision of genetic material into the everyday life of our hero, they effectively swap identities after a little bit of physical change. And our hero, John, becomes Jerome, and he is now a valid. So he goes into the space program, and he does magnificently well. And I won't give away the end. Uma Thurman, for those of you who weren't interested in Ethan Hawke or Jude Law. Uma. And so he eventually gets shot into space, perhaps, and gets the girl. So this is a pretty strange scenario. Children having IVF, genetic testing on embryos, selection of genetic testing on embryos, DNA sequence determined at birth. We look at that sequence and we can work out some characteristics, and then people then choose paths in their lives, or they have them chosen for them in this case, on the basis of their genomes. So is this science fiction or science faction? Before I go to that, this is actually my favourite scene in the movie. It's hard to see for you, but this is a young lady who's at a nightclub, and she's put her head here through a small hole, and on the other side is a technician who's got a Q-tip, as the Americans call them, a little cotton bud, and she's just kissed a potential boyfriend. And so he's getting the DNA from her lips and sequencing it, and 5 minutes later she gets a printout, and the comment goes something like, "Oh, you've got a live one here, honey." Science fiction. Maybe. So let's go back to today. Science fiction or science faction? We've already heard from the great talks from Doug and Vanessa that a lot of technologies are around us. For those of you remember the first scene from this movie? So this is what happens to every child born in Australia, probably for more or less the last 30 years. For those of you who are around about 30 or less, somewhere in a hospital in this country is a piece of filter paper with a blood spot on it, and you have all been already diagnosed for a minimum of 5 genetic diseases. Every child born in this country, either at home birth usually, or within the system, is automatically tested. So somewhere for all of us is either blood, or most of us, is either blood or DNA sitting there, and we have already been tested for 5 genetic diseases, and my clinical colleagues in the genetics departments tell me that could be up to 27 different strictly genetic conditions. So this notion that we're going to live in some society where every child is genetically tested at birth, well, that's preposterous, right? We already live in it. We already do it. That's okay. So, then we've already heard, this is the 3500 ABI sequencer. There's a plug for the alternate manufacturer later on. So this is what the machine looks like, and as we've already heard beautifully, and I won't belabor it from both Doug and Vanessa, we have I think about 10 of these up in Queensland at the moment in one room. They can do about 20 gigabases a day, and that's in terms of human genomes, that's about in raw data, that's about 60 genomes a day. So theoretically we could produce enough data, although as Vanessa said, how we could analyse it, I don't know, for 600 human genomes a day. And Doug already showed this slide showing that the cost of a human genome sequence is going down. So I won't go over it again, except to say that last week I got an email from a Chinese company offering to sequence not just my DNA, but any DNA I want to send them, a complete human genome, send them the DNA. They send back the lot, $4,000. Now, getting pretty close to that MRI scan cost of $1,000. So It's going to happen. It's already happened. We've already got people turning up to their GPs with their genome sequence. Okay, so what can we do with it? Once again, as both Doug and Vanessa have mentioned, this is a piece of genome sequence, and the little yellow bases here are those that vary between individuals. And we've heard some beautiful examples of that today already. So this sequence is about 1/1,000,000th of the genome, or as I put it in terms of a story, since this tells a story, less than one page of 2,500 John Grisham novels. He seems to write them at 270 pages, which is perfect for a trip from here to Singapore or something like that. So we vary around about 1 in every 300 of these bases, and these small variations between us and our interaction with the environment are what make us what we are. So how can the science of Gattaca, this idea that we can predict not just strict diseases, but characteristics about all of us, how is that finally going to be realized? Well, we've seen over the last 5 years a snowball, an amazing avalanche of data, which is suggesting that we're beginning to understand how these little changes affect our risks. Actually, parenthetically, if I were to change, I think if I were to make, this is a section of chromosome 9, if I were to make about 30 specific changes on this sheet, this would go from being me to being a dolphin. So we're actually quite related. So all I've done now is grab from a very learned journal, Nature Genetics, and another one, a couple of screen grabs of recent articles over the last 3 months, just to give you a flavour for the sorts of things. So these small variations are called single nucleotide polymorphisms, and the idea is to try to work out whether any of these small innocent variations, 1 of them, 5 of them, 10 of them, 50 of them, all can aggregate together to increase or decrease our disease risk. And as we go, so we can see variants at some genes are associated with Alzheimer's disease risk. There's a suite of about 11 of those single changes now, or I think it was actually about 15, that ultimately when you put them together, you can come to the conclusion that somebody with those 15 changes is 11 times more likely to develop Alzheimer's disease than somebody without them. So that data's out there. So, and just about any disease, the data are beginning to be aggregated now. Many studies are being put together. And as Vanessa raised, it's very important to do this in different populations, and so those data are just beginning to come. And so here are variations associated with the risk of, say, endometrial cancer, and I won't go on and on. Coronary artery heart disease. Now, if you have this set of changes, I have to emphasize, it does not mean that you will get coronary artery heart disease. It gives a risk. You may be 2-fold increased, or 5-fold increased, or 10-fold increased. It doesn't mean that you will get that disease. So, and it's not just diseases. Or there's colon cancer, and just about any disease you can think of is undergoing this right now. And it's not just diseases, it's conditions that are seen to be somewhat unusual in the population, things like narcolepsy. There are variations now associated with restless leg syndrome, which is a neurological condition, and it goes on and on and on. It's things that happen to us as we age. So this is looking at the age of onset of menarche. And so, and it's just not just the way we have, whether we have diseases, it's not just the way we age, it's perhaps the way we look. It's changes in our DNA which look at our waist-hip ratio and the way our fat's going to be distributed, which of course can impact on disease as well. It can be with, this is from some colleagues in Queensland, it can be whether you have straight hair or curly hair. And it can even be into behavioural traits, such as smoking behaviour, variations in the DNA that mean that you are X times more likely to exhibit a certain type of smoking behaviour than anybody else. So, and finally, it can be all for, this is from a former colleague of mine in London, and Bob Williamson, who's sitting down there, Steve Humphreys. This is hard to see, but the blue bar is the distribution in the population of one of these simple genetic changes. The red bar is the distribution of a very simple genetic change in people who are elite athletes. Now, in fact, as it turns out, that people who have a very simple genetic change, this one, if you look at people who have ascended over, I think it's either 6,000 or 7,000 meters, so mountain climbers, those are the very elite mountain climbers, there has never been, the only people who've managed to ascend over 7,000 meters have been people like this. So, and if you take people who are like this and you weight train them, if you look at the increase in muscle mass, they train a lot better as well. So everything from our disease risk to the way we look, to perhaps the way we behave, to whether we should be selected for the Australian Academy of Sport based on our DNA. I mean, why would you take somebody into your elite mountaineering program who did not have this change? Really? I don't know. So, where's this all heading? So this is an iPhone app. No, this is an iPhone app. Vanessa's already showed us a very nice example of this as well. This is my genome. It comes out of the company, one of the sequencing companies, Illumina. It's not quite released yet, so the idea is that you carry around your genome. So I'm now not at birth. I'm now with consenting adults. Carry around your genome, and you upload it to your doctor. Upload it, and away we go. Then this is the CEO of Illumina, and he's put his genome out there. And you can go through using that sort of genetic information I just showed you. You can split it down into traits, curly hair, blue eyes, however it goes, obnoxious bore at dinner party, that sort of thing. Disease risk and pharmacogenomics. And the interesting one here at the bottom is ancestry, which I think is actually an amazing little thing because people now are getting their genomes sequenced, they're uploading them to the web, and if you give permission for them to be made publicly available, there are programs now that will tell you whether you are related to other people who have uploaded their genomes to the web. So rather than ancestry.com, you just put your genome up to the web, and then a program tells you, oh, you've got a third cousin living in Sydney. So that's what that button is. And so when we have a look through here, I'm almost at the end now. Hold it. Oh, I think this has died. There we go. And we have a look here under pharmacogenomics on the basis of Jay's DNA. It says that he should probably, as far as his statin, his cholesterol problem, he should probably using this one. It's the best one, and the worst one is lovastatin, and this is the dose you should have. Okay. So the thing is, we've already seen that the Gattaca scenario, the IVF technology, we have most of it. Sequencing is getting faster and cheaper. So the notion that we could be tested at birth is possible. The notion that on the basis of simple changes in our DNA, that we could predict traits, call them what you want, not necessarily diseases, but the way we are, the way we act, the way we look, that looks likely. And broadly, as an ethical question, you might think, well, I don't think we should be testing everybody at birth genetically. Well, we already do it. Now, we've been involved recently in discussions with the Commonwealth Government about how to take genome information and apply it to the community for the better health of the community. And of course, the easiest one because of ethics is people with diseases like cancer. Take a tumour, sequence it, do our best to work out what's going on, and then feed that information back to that individual. And perhaps there's a drug that will be useful that we didn't know about. That's, you could see how that's a pretty simple question. Very hard to do right now, still very hard to do. Where the ethical debate really gets interesting is when they take the notion of whole genome sequencing like Gattaca and apply it at birth. We already do this. So the question is, are we really going to take it that next step? Well, technically we can do it. So I think history shows us that as time evolves, our ethical frameworks clearly evolve. And I think at the end, just in my final comment, I think that we are currently all waiting for government and people like Doug and Vanessa, and we all sit in a room and we talk about how this could be actioned and how we could do it. But the reason I showed you that iPhone app is that it is being taken out of our hands by companies. I can send my DNA, as we've already seen, sequence, done, and people turn up at the doctor with it. The next step is to send the DNA of your newborn. It's not illegal. So while we sit there and talk about the ethical framework of how we're going to deliver genomic medicine at a population level, I think we're going to wake up one day and find out it's here. And the reason for that is that private enterprise is already doing that for us. Thank you.
Thank you very much, Brandon. So we've had a feast of challenging ideas, and we've seen a challenging future. It's over to you now. And we'd really like to have your questions and have a robust conversation with our three speakers. So who's going to be first? We have one here, and we've got a roving microscope and one up the back, so I can see three to start with. First, second, third. Thank you. If you could pass along the microscope to the, maybe you could, sorry. Sorry. I need to get back in the lab, yes. Please introduce yourself and then state your questions succinctly, and we'll want succinct answers too, but go for it.
Thank you very much for those very stimulating talks. My name is Liz Burton, and my question is, when you look at programs by David Attenborough, looking at herds of animals, different species, the entitlement to breed tends to be fought out by the dominant males in a population. And they fight it out, and one recedes or is
You've been
damaged.
watching the apes program on 7:30 on Channel 2, yes.
Well, a variety of species, they do fight it out. Now, here with humans, we don't do that. Everyone's entitled to breed. But I'm just wondering, when you project forward some of the ideas that are being advanced, that we could be breeding a warrior species due to the attributes that we admire with those qualities. And if we do that, if we do inadvertently create a warrior breed, are we ultimately going to be rather counterproductive to the goals of our own species? That's the question.
A very interesting question. Who wants to tackle that one? Oh, we've got Vanessa first and then Brandon.
No, I'm pointing to Vanessa.
Is this on?
Yeah.
Yes,
Can you hear
it
me?
is on.
Okay. So to answer your question, actually, we can answer it already. We are breeding a warrior versus a warrior species. So when we look at the Kusan people, the hunter-gatherers, we do know certain alleles when we talk of alleles, genetic codes, you heard Brandon talk about those single nucleotide polymorphisms that have been associated with warrior status. What we mean by those who don't know the difference, a warrior person is someone who will react instinctively to a stimulus. So to be a hunter-gatherer and to survive as a hunter-gatherer, you have to react. So if you see a lion coming towards you, and if you're going to think and wonder, now, what should I do? Should I go this way? Should I go? You're dead. You've eaten. So it's natural selection in that case. In the past, we needed to be warriors, not warriors. A warrior is someone who has to think about it and decide. And as Eurasians, Europeans and Asians, we are more warriors. We want to think about everything. But then that is natural selection, it's natural change that happens. We just had a debate, we had time to kill. We were 10 of us from JSVI on a long road trip, so we were discussing how the world is changing. How there's more Asperger's syndrome seems to be present. Is that because there's a place for people with Asperger's in this world? Is that a good thing or a bad thing? That is natural selection at play. Okay. It doesn't really answer your question, but it is natural selection is happening all around us, whether you want to rate that as good as bad. Remember, compared to animal species, we are very different. We have populated the globe. We have changed the face of who we are, whereas other species didn't do that. We should still be hunter and gatherers, but we're not. We changed that 10 to 12,000 years ago. We have to live with the new world we created.
It seems to me that this conversation will be enriched if we also give the opportunity to the audience not only to to ask questions, but also to comment on the questions. So before we move to the next question, is there anyone in the audience who might like to add to this particular conversation? I was wondering if Matt would like to. Yes, could we have a microphone just quickly down the front here? Thank you.
Yeah, I mean, my view is that human beings at some point went through a period when they selected very strongly against the kind of impulsive aggression that, which I think is the point that Vanessa was making, and that you know, we tamed ourselves rather brutally by clobbering people who were too aggressive or locking them away or something. And to some extent, we're still doing that. I mean, people who are too impulsively aggressive end up in jail and don't have as many babies as people who are not, I think. So I wouldn't be worried that we're getting more aggressive as a species. I would think we've been heading in the other direction, particularly in those dense societies, you know, of South Asia and so on, where you have to get along with your neighbours because we've been living densely alongside each other for thousands of years. So we've on the whole been taming ourselves, I think.
Any further comments? I could take one more on this particular question.
Could I comment on that as well?
Yes.
You know, I wonder if that desire to mate with multiple partners is less to do with aggression and more to do with status. If you think of blues musicians and artists and things like that, you know, maybe the traits that need to be selected for, if that sort of dystopia is the one you want, are very different from raw aggression. So, you know, maybe it is creativity or status or whatever else. Unfortunately,
last comment on
scientists
the
don't
question.
have those qualities, at least in my experience, Brandon. Maybe a man of your magnetism, but
Wrong guy.
I won't comment. Yes, final comment on this question.
Yes, Galina Hitchin. We've already got skewing, and that is amongst sperm donors to IVF programs. It's not a cross-population spectrum at all. It's certain kinds of men who are donating sperm for IVF programs, and in some cases in the US I've heard there's bias towards such and such who must be, you know, Nobel Prize winning and tall and blonde and blue-eyed and so on. So we've got a lot of skewing already happening out there with unknown consequences.
Fortunately, that's not a huge percentage of the population. Sorry. Now, our second question was somewhere, I've forgotten. Thank you, yes. Please introduce yourself.
Hi, my name's Julie Egan. We started talking about the human genome and now we talk about our individual genomes. But I wonder, what percentage of our sequences do we share? So what percentage make us human and what percentage makes us individual humans? And I'm wondering whether knowing what is common to all of us has contributed to the cost of driving down the individual sequencing.
I think that's a question for Vanessa too.
Yeah, we're about 3% unique as individuals, and that was actually, with the Human Genome Project, slightly higher than was anticipated in the beginning. So we found out that actually from the Human Genome Project, probably one of the most significant findings, just taking the genome, was that we were more unique as individuals than we thought. But still, we are one species. There isn't a lot of difference, but that difference we need to understand. The out of Africa component obviously is a lot smaller. If we just, it's easier for people to understand that mother's variation that we inherit, which is only 16,500 bases. In that 16,500, if there are two Europeans sitting in the room now today, and you look at the mate on either side of you, they will have 20 differences. If you were two Bushmen or Kushan people sitting next to each other, you would have around 85 differences between you. So it doesn't sound like a lot in 16,500, but those differences are critical.
Okay. I think that was a question of fact, so probably not needing discussion. But so is this a different question? But we've got one more. Yes, we've got one more question ahead of you, and then we'll come to you. There's one up the back in yellow.
Thank you. My name is Helga Kuse, and my question is to Professor Hayes. So we can trace our DNA back to our African inheritance. My question is, how far back can we go? Could we, for example, still detect chimpanzees' DNA in our genome?
So, yes, chimpanzee was one of the first complete genomes to be done for this reason, because it is our closest relative. And that's where we get the time frame. So we use what we call molecular clocks to try and trace back what was a common ancestor. And I think that drawing that you always see from chimp and we grow, we crawled and we came up and now we modern humans, and of course we've gone down again because we sit in front of computers the whole day. We've forgotten how to walk and we've forgotten how to talk too, is misleading, because we weren't a chimp. We had a common ancestor with a chimp. And that's very important to understand. We don't know what that common ancestor was. Because we know how DNA changes over time, we use that knowledge to infer a time frame when that common ancestor must have lived. And that is between 6 and 7 million years. But we got to realize that these tools are based on our knowledge, and we have to, with these tools, put in what we think. So they're not exact calculations. We will, over time, start learning. Going back in time to actual fossils is really hard. I mean, one of the big questions I always get is, are we going to bring back dinosaurs? For those who saw the Catalyst program when the mammoth was sequenced, for example, everyone's question wasn't about, gee, this was great to sequence the mammoth, was, when are you going to bring back dinosaurs? Probably not, they're too old. So what happens is, with time, the DNA breaks up into really, really small little pieces, and it's hard to piece those together. And especially when we're looking at humans, this was the problem with sequencing Neanderthal, and the Neanderthal that was sequenced was between 40 and 50,000 years. So we know that's about as far back as we've gone. The mammoth was 60,000 years, but that was easier. The mammoth was in a permafrost, so it was stored, the DNA was stored a lot better. The biggest problem with trying to define who we are as humans and going back to our homin cousins and even further back down the tree of all the homins that used to walk with this earth, remember there were many, we are the survivors of all those homins that used to walk the earth, is that we naturally contaminate that material. So it was easier with the mammoth because as a human we can't contaminate a mammoth. But to look at, for example, a homin species, we don't know how much of that is actually ourselves. We're looking at the people in the lab working. People find bones, they touch it with their hands. Your bone is very, very porous. Immediately, you're transferring the cells from your hand, like in Gattaca, straight onto that bone, and it's really hard to decontaminate because we don't have a control. So we use the chimp as one of those controls as well.
I think Doug had a comment.
I think one of the most amazing discoveries of the 1980s and 90s was the realisation not that we share pieces of DNA in common with chimps, recent, from whom we've more recently diverged, but that some of the fundamental genes that regulate things like body patterning or vision are shared with common ancestors that we had with fruit flies and before them bacteria. So, a lot of our DNA is made up of very similar blocks to much more primitive organisms. I think that was one of the great realisations.
Exactly.
Maybe I could make a comment here. I fell in love with DNA when I was a student at this university, and it transformed my life's journey. And I'm still entranced by DNA. And the idea that blows me away the most is just that my DNA bears the evolutionary marks of organisms of the very simplest kind, so that I am related to everything in this universe plant, animal, bacteria. It's a mind-blowing thought, and it gives you great respect, I think, for the diversity of the world and why we should preserve it.
Maybe we could say it's a whole field now called comparative genomics, where we take all organisms that we know that have been sequenced and we compare them because we learn from each other.
Yeah. Now, yes, we have another question. We've got all the women asking the questions this morning. I'm glad you're asking a question next, yes. Can we have a microphone down here, please?
Thank you for a wonderful trio of talks. My name is Doreen Rosenthal, and as a social scientist, I'd like to shift the focus a little bit with my question. The science that you've described is fascinating and somewhat frightening. And Brandon talked a little bit about the ethical implications of having an app on your phone and going to your GP. My question, I guess, is how do you get people to be willing to do that when we know, for example, that many, many people refuse to be tested for various disorders? In particular, I'm thinking of the BRCA gene, not just because of their worries about insurance, but because they just don't want to know. So you're talking about taking your app to the GP and finding out that you have a 98% chance of this disorder or that other. How do we marry, in a way, the science with people?
Brandon, would you like to answer that, and then Doug can comment?
Well, I don't think we'll ever really, ever's a terrible word to use. I don't really see it as actually encouraging or forcing people to get their genomes done. I think in the practical sense, the scenario described is that people will still go to their GPs with a disease, and then the GP will boil it down to be just another medical test. So there's a difference between this prospective, the idea that I might be at risk, and go to my GP. That, in a sense, I think that what will happen is that the GPs will do the right thing and explain that having your DNA tested in particular areas is actually no different to having cholesterol measured. High cholesterol is a biomarker, is a marker that has a test, it has a low score and a high score, and there's a bit of a guess is to say if it's in this range, you're at risk for heart disease. And so they're just going to shift the test. So I see I see these things in very simple terms, that it's just another test. And I think when you start talking about it as being individual tests rather than somebody's complete genetic identity, I think that's the way it'll start biting into the populations.
Doug, did you want to add to that?
I've always been actually impressed by the opposite tendencies, and that is where groups that are perhaps superficially conservative have actually embraced these sorts of technologies to solve a particular problem. And it's really a cost and benefit analysis. And the two that I've read a little bit about for a couple of articles that I've read around the Ashkenazi Jewish population who had a very high frequency of Tay-Sachs disease, which was an absolutely horrific genetic illness that kills, where the kids develop relatively normally and then decline in a horrible way, and that genetic testing among couples that were contemplating marriage was something that was really encouraged by that population, and it's been expanded from that single disease to a number of diseases. Likewise, a situation, for example, in the Mediterranean, in Cyprus, where particular blood disorders were costing the country a fortune, and if there wasn't a genetic intervention, basically to treat the kids that had those disorders would occupy the vast majority of the healthcare budget. And again, the Greek Orthodox Church in that case was very pro these types of tests. So for me, I think it is a sensible explanation of the risks and the benefits of the technology. And I've found, especially in Australia, once the benefits of a technology are apparent and explained in a matter-of-fact way rather than foisted on people, that technologies are actually taken up very readily.
I mean, Bob could probably talk about that. I see he's got his hand up because he was part of the
thalassemia
implementation
project,
team
yeah.
of the thalassemia project.
This is Bob Williamson.
Bob Williamson, University of Melbourne. I think that particularly in a university, we should remember that the underlying philosophical position is that knowledge empowers. And that doesn't mean knowledge comes without a price. That doesn't mean that knowledge is always welcome. And nonetheless, at the end of the day, it's ignorance that deprives people of the ability to do things, and it's knowledge that empowers them and gives them the ability to do things.
It gives them a choice.
And I think we really have to say that doesn't mean people have to be forced to take knowledge, but by and large, it's a good thing when knowledge is out there. And in the examples that have been given, thalassemia in not just Cyprus, but also in Greece and Southeast Asia and so on, where something can clearly be done to make things better for the community or for the individual. I mean, hemochromatosis is an obvious example where genetic testing is embraced by everyone who has the test. Because if you are going to develop hemochromatosis, all you need to do is to give blood every 3 months and you don't develop hemochromatosis. You don't even have to take drugs, and it's fabulous. I think the issues come where it's not clear-cut exactly what you're going to do. And I just caution people, we'll be discussing this again, I should say, at 3:30 this afternoon. But I should give you one example. There's a very good predictive test that tells you whether you're at high risk or low risk of developing Alzheimer's disease when you're in your 70s. And this is an ApoE4 test. And by and large, doctors say, oh, we shouldn't offer this test because you can't do anything about it. But by that, they mean you can't do anything medical about it. But many people who are in their 60s or 70s actually want to have the test because there are life choices that can be made that are actually quite useful from the point of view of the individual if they know whether they're at high or low risk. But I come back to the point I made at the beginning. It's knowledge that empowers people. It's knowledge that empowers a community. Ignorance disempowers, and we should always, I think, defend that principle.
Thank you.
Can I raise...
Sorry, yes. And then we'll have Lauren Skeen from the back.
I thought I'd raise, because you mentioned the word social. So the examples that we've just addressed, which are the most, probably the most relevant at the moment, is a direct disease association. But there is also social impact why some people have their genome sequenced. And obviously the two that I mentioned this morning, one being the Archbishop Desmond Tutu. He was advised by his own advisory board not to have his genome sequenced. Now this is very interesting that people often want to protect other people because they feel that they can. And he turned around and he said, I want to be sequenced because I know that by putting my sequence out there, I'm going to make this technology available for my people who never had these advantages. He was not afraid that we would find anything that would impact his life. Yet, we did find something which has dramatically impacted his life, but he never believed that we would, and it wasn't a disease thing that we found. We found that he had an ancestry to the Khoisan people, which he never knew. And what they did for him was when he met the wife of the gentleman that we sequenced, Khoisan individual we sequenced, she reminded him so much of his passed away mother that he couldn't stop hugging her and embracing her. And because he grew up in apartheid and fought apartheid, he was not given an identity by the South African government when he was sent to Cambridge to do his theology studies. His travel document, he wasn't given a passport, said, identity unable to be determined. So he turned around, and it was Ronald Reagan who finally listened to him by pulling out of his pocket. He was frustrated. He could not get through to Ronald Reagan that apartheid was a problem. So he pulled out his travel document and said, look what I'm being called. And then for the first time, Ronald Reagan reacted. And he said, now I can say, if that government still existed, which it doesn't, those people have passed away, that you didn't want to give me an identity, but I'm truly a person from this earth, from this place, that you wouldn't give me an identity. And that social impact was we didn't think that would happen. And for the Kusan speaker, he knew clearly what he wanted. He's not going to go to a medical doctor, never been to a Western doctor in his life, has no desire to ever go to one. But what did he want from it? He wanted his children and his grandchildren to be proud of who they were. Simple. And he believed by scientists telling the world exactly what he knows. He knew exactly the importance. He knows they've been around forever. But he said the rest of the world will now recognize us, and we don't have to be ashamed of clicking. We don't have to be ashamed of who we are. So you can actually empower people through ways that you never thought was possible. Just thought I'd throw that one out there.
Yeah, that's a very interesting angle. Lauren, yes?
Thank you. I've been enjoying the discussion very much too. I'm Lauren Skeen and I'm a lawyer. Just wanted to pick up something that Doreen said, and it's with regard to insurance. So you have people saying, well, you don't want to have genetic tests because you won't be able to get insurance. Now, in Australia, we have community rating for all of our health insurance, both public and private, and it doesn't depend on risk. So you always get health insurance. What people are worried about is life insurance and other income protection insurance. And this does vary with risk. So if we're really worried about this, one of the things that we could do is adopt a policy that you can get an insurance policy up to a certain amount that doesn't depend on risk. So this is one of the policy options we could be discussing. But also mentioning something that Bob said about hemochromatosis. This was the first condition where life insurance and income protection insurers were prepared to not impose an added cost for hemochromatosis because they could see that there was an effective intervention. And so if somebody has hemochromatosis and they reveal that to an insurance company, they can still get life and income protection insurance without paying a higher premium. Now, I think as knowledge expands, we're going to find that there are more and more conditions like this. And so even with those types of insurance, it won't be a problem. But the main thing to remember is that health insurance for us in Australia is available to everyone.
Thank you. I think those are very important facts to know. Yes. Is this a new question or a comment on that last one? A new question. Just here in the white shirt. No? Further forward. Yes.
Thank you very much. Mark Hargraves. I work at the University of Melbourne. I'm a physiologist. And I wonder if sometimes the technology has moved too far ahead of the physiology. I think there are lots of examples where there are well-defined genetic bases of diseases. But in many instances where there is perhaps a polygenic basis to the disease, and many of the high-frequency lifestyle diseases are determined by multiple gene environment interactions. And perhaps, as Doug said, the benefit of the technology is that it provides the scientists with the kit bag that they need to understand the biology better, and that one of the potential downfalls of the high technology and the low cost is that lots of individuals may be trying to make decisions on knowledge that isn't fully complete. Recently, not surprisingly, some of the complex diseases such as diabetes are better predicted by physiological characteristics, some of the biomarkers, than which probably culminate or are the culmination of quite complex multiple gene, multiple environment interactions. So,
I think another point to take from an extension of what you're saying is it's going to be absolutely no good at all having our DNA sequence on our app until we have much more knowledge in terms of what the genes actually are telling us in terms of complex diseases. But B, we've got doctors who are across all of these things and can wisely advise the people coming to them. So there's a long way to go. I think what we're doing is pointing to where it's going to be in an ideal world. But there's a lot of work to do and a lot of knowledge to accumulate before we get to that position and are able to use it wisely.
Could I make a comment on that? I think we've been through this before, though. You know, if you wind the clock back again 20 years, molecular biology was all the rage. Certainly in institutes, we were desperate to hire molecular biologists because we thought that, you know, by understanding DNA and being able to do recombinant DNA will solve all of the tricks. Very quickly became apparent that what we needed in order to put that into context were people that deeply understood biological systems. You know, I think we're going through the same thing with molecular taxonomy and you know, in our museums, that there's you know, a hope that we'll be able to understand evolution fully by looking at just the DNA. And at the moment, there's a trend to, you know, make classic taxonomists redundant. You know, within a year or two, there's going to be a realisation that what we need is to embrace technology, allow us to be able to use those new tools, but to do it in the context of a deep understanding of the physiology and the biology of the organisms that we're studying. So I think it's just the same pattern repeating itself. And I know as director of an institute, you know, the laboratory heads that I'm really interested in recruiting are the people that have a deep, abiding passion for a particular underlying biological process or tissue, because I think they're the people that are going to put the information into the best context.
Brandon, you wanted to add to that?
It's certainly true that this DNA variation, excuse me, information that's out there now is really needs meta-analyses, you know, the same thing done again and again and again. And so the real problem that the personalised genomics field has is actually the companies, in the sense that I think 23andMe and some of the others, if you drill through them, they all use different SNPs and different predictions and different, they all give different information on the same DNA content. And so there's no regulation, and, you know, we're a long way away from having...
Using it wisely.
Oh,
yes.
From using it appropriately, a
Yes.
long way away.
Yes.
Maybe this also, following on what Brandon said, one of the big topics we had at our conference at the beginning of this year to look forward in the next 10 years, and also to look at, we look a lot, and there's been a lot of publications to mark the 10 years since the human genome on what the achievements were, but very few conversations about our failures. Not really failures, but one of the big ones that come up is, have we sufficiently associated phenotype with genotype? So with our DNA information, and we haven't. And so we need to take a step backwards, and we have with certain diseases, but with the complex diseases we haven't, because we need to take a step backwards and say, we need to work, as scientists, we've moved into a completely new era where we don't work in silos anymore. We have to work across the disciplines to truly understand the disease. So we need the clinicians to work together with us to define that phenotype and what we're putting in. One of the diseases I work on, someone mentioned earlier, breast cancer, but for example, prostate cancer. What is prostate cancer? It's probably disease A, B, C, D, E, but we're just putting into our algorithms prostate cancer. So we need to be able to phenotype much better.
Strategize. Yes, question here. We've got 11 minutes left.
Bob Phelps from Genethics.
Sorry, yes, and then this one done here.
Thank you. In her closing remarks, Vanessa mentioned the synthetic biology enterprise of the Venter Institute, just like a throwaway, that you've got an organism, a new organism that never existed before, 1 million base pairs, still alive, purpose to design vaccines and biofuels. This is a commercial enterprise like most of this science and technology, and I think we need to hark back to what's driving it, and it is dollars in the end. In the Gattaca example, we were assured that many of these things will be just another test, that they'll be normalised. But we already have the heel prick test, which all infants have in Australia. Those samples are kept on file. Who owns and controls them? That's a serious issue that is never addressed. They've been kept now for some 30 or 40 years. We're all on file, those of us who are younger than that age. And our pathology samples, similarly, are a potential commercial product that are taken and owned by other people whenever we give a sample. So I think these social context questions need to be addressed as well. And I'd like to hear how Venter in particular is addressing them.
Yep. Okay, so the J. Craig Venter Institute is a non-for-profit organization, and everything as if the human genome and anything to do with genome sequencing now has to be released freely onto the internet, to the databases. Otherwise, we won't exist. NIH will shut us down tomorrow. So this information has to be made freely available. As far as the synthetic biology is concerned, there is a private company which is not part of J. Craig Venter Institute, Synthetic Biology, and that is funded by a fuel company. And they realized, Exxon, they realized very early on that this planet is running out of fuel. It's a plain and simple problem. We are running out of fuel. And so we have to look forward, and of course this is an expensive process to discover and be able to generate an alternative means of fuel where we're not taking out of our planet. So the Venter Institute is dedicated to zero carbon and to saving this planet. So much so that we're building our new building now, which is going to be the first zero carbon laboratory in the world. We will not draw energy from anything else but generate it ourselves. Because we're trying to use this as show it as an example to the rest of the world, that this is a real issue. And we can't address it when it's too late like we normally do. We have to address it up front. But yes, of course, that needs dollars. And that's why there is a separate institute, Synthetic Biology, which is directly involved with generating that, the synthetic. But the research arm of what we do, everything has to be released. So all the new microbes discovered in the sea, everything was released. Everything in the gut, everything's freely available for the scientific community to look at and use.
So, Vanessa probably won't thank me for this, so I apologise now. But I can say it, but Vanessa can't probably. Her boss, Craig Venter, is not only a brilliant scientist, but a real, larger-than-life character who's just, he's a sort of modern Jacques Cousteau, really, isn't he, the way he sees himself as going through. And he's absolutely brilliant. And in the press conference, I believe, for the synthetic life form, he said something like, when people accuse us of playing God, I think about that and I say, we're not playing. So,
don't say that.
But
he didn't get the gene for turn it off.
I know, that's true. But he's a showman as well.
Back to legal, ethical and moral dimensions, let me remind you that in fact this afternoon at 3:30 there will be another workshop and discussion panel on these very important issues, and I encourage you all to attend that. Bob Williamson will be chairing that. Maybe I could throw one, so there's one question at the back, but before I ask you your question, seems to me we had a question before that touched on an area that does deserve some discussion, and that is, are we in danger of exerting our own selection on our offspring and running the risk of minimising our genetic inheritance? Because I was thinking of Gattaca, so I'll ask you to comment on that, Brandon. If we actually, how will we not allow too much selection to go on in an ignorant fashion about just because people want to have a child with blue eyes and who's an athlete, for example?
I mean, that's a very good point, Suzanne, to which I don't have an answer. I think it is a real risk. People, so, I'm tempted to say if we let people get in that situation, but I just have the feeling that it won't be a question of us letting, it'll be a question of people doing. That's my unease at the whole situation. So, I mean, I can only agree with you that that's a potential problem. I think it's a risk,
and that's why I think that actually much more knowledge about genetics is really critical, because then hopefully people will understand the complexity of genetics and how by selecting for a few single attributes, they might be running risks in other directions and ignoring other things that are very important for their children as well. So I think that a much greater understanding and familiarity and deep understanding of genetics is really important across the entire community.
There's a young chap there who's been very, very patient.
Sorry.
And you've missed him, and he's looking aggrieved.
Suzanne, maybe I can approach that question.
You've not forgotten, and neither are you.
So one of the fears, obviously, with synthetic biology is that we're going to take it even a step further and generate synthetically the ideal child or offspring. And obviously, this is a lot of comments that came out after the synthetic biology paper was published. So we scientists have to take the responsibility of what we're doing. And that's why when the synthetic biology concept was started 15 years ago, the Institute started immediately with ethics and policy development and got on board a whole ethics and policy group. And that's why it actually was taken incredibly well in the United States. Even the Pope congratulated Craig Venter. And why was that? Because the community was not shocked by the time it came out, because they had been pre-warned that this was, and there was pre-discussions going on for 15 years, that the American public actually voted more for it than against it, which was quite a shock. So the Obama government had discussions prior to even the announcement with Craig, with the policy team, to make sure, and we have to take that responsibility, because yes, of course, there is the fear, and of course one cannot control, but you try put the policies out there, you try put the, there is in every genome that we generate, there is a switch that can destroy it, for example. There is these, but of course you cannot control what someone else does. Or you can say at this time, it took 15 years to do it, so if someone manages to do it at that level in a lab, you know, that this is going to be a huge task. It certainly can't be done at this stage by an individual. So yes, we do have to address these issues, but we have to address them at the beginning, not when we're finished.
Thank you, Vanessa. We're running out of time, so I want to be quick with these questions.
Hi, I'm Sam McQueen. You've touched a bit on commercialization and sort of the commercial side of modern genetics. Vanessa, you said how the Human Genome Project, all the data generated from that is in the public domain. But identification of genes has been done by commercial enterprises, and genes are being patented, and that has great repercussions for medical research and also for legal and ethical dimensions. I'm just wondering what you all think of the potential challenges in terms of the law, ethics, and health research for the commercialization and potential commodification of genes and genomes.
Well, I hate to say, but that is the topic of this afternoon's session. So please come back to that session, because I think it's too long a conversation to have right here, if you don't mind. And I'm sure that there'll be many, many of those questions addressed this afternoon. I think our last question's up the back.
Just at the front there,
sorry,
Suzanne.
There's another one here. 2 last questions. It's very hard to see everywhere at once.
Am I first?
Yes, you're first.
I'm a student of biochemistry and genetics. I've noticed that it's been a recurring theme in these talks that the private industry has been leading the charge with these technologies and their application. The last speaker repeatedly used the argument that we're already doing it, so what's the problem? I'd just like to ask the panel if they think it wise to hand over control or relinquish control of our ethical evolution as a people to the dollar-bound hands of the corporate entity.
Well,
can I pick that up?
Yes,
Just
but
at the
I
end.
think you should point out that there are many controls exerted on scientists by government regulations, for example, with NH and MRC that deal with these issues. So it isn't a totally free enterprise at all. Brandon?
Yes, and I really, I don't think it's really quite an accurate summary my sentiment is not the private industry are doing it, so what's the problem? It's almost private industry are doing it, so perhaps we're going to get a problem, because we as scientists working in the public domain are constrained by the legislation and debate and the legislation of this country, right? many of the scientists who are contributing into the private domain are certainly not constrained by our legislation and are in our country. So they're on the internet. So, you know, I agree, it's a problem. But we also have to admit that, you know, regulation only gets us so far because, you know, in this global world, there's only so much we can do. So we just have to prepare for the consequences.
And we have to make people, as individuals, aware of these issues and hope that with knowledge and wisdom, the community pressure will be brought to bear on them anyway, if it's not brought on them from inside as individuals.
I'd also like to say, you know, I don't think it's all downside having private capital involved in making the very large investments that take advantage of these things. You know, I think a lot of the new cancer therapies which are targeted to individuals' genetic changes in their tumours require hundreds of millions and billions of dollars to get from the basic discovery in the lab to a pharmaceutical that's capable of being used by the general public in a hospital. You know, we have a large enough problem in Australia managing the economy of the healthcare sector at the moment, let alone having the government invest in speculative drug discovery exercises. So, I'm really upbeat about the advances that will make a difference in my life and my kids' life that have come from well-regulated private sector investment. So I don't think it is private sector, evil, society somehow omnipotent and capable of walking a wonderful ethical path.
I'll just completely reaffirm Doug's point. So many of the things that we deliver to the community are delivered via private sector because they're the ones that have the investment. And the commercialisation is to be welcomed.
And I also have to say that private sector actually can't function without us, and we can't function without them. So most major breakthroughs are done as partnerships.
I think that's a very good point to finish on. I've been told that we must finish at 12. It's already 12:02, so I'm not a very good chairperson. I'd like to ask you to thank all of our speakers and panel members.